Take your seats. So welcome back. The next talk will be given by Wendy Hwang-Verslues from Taiwan.
Thank you. Can everyone hear me? All right. So thanks for the opportunity. It's really great to be able to tell you what we have been doing in the lab. So I would like to start my talk by going through the breast cancer progression. In the normal breast duct, it consists of a single layer of luminal cells surrounded by a single layer of myoepithelial cell within the basement membrane. When cancer lesions start, the cancer cells will proliferate in the duct, and it will continue to proliferate to form the solid tumor. And the center of the solid tumor will undergo necrosis because of lack of oxygen and nutrients. But the cells surrounding this area are under tremendous hypoxic stress. And now we all know that hypoxia is a driving force for metastasis, which allow the cells to acquire ability to break through the basement membrane, interact with the endothelial cells, form angiogenesis, or intravasate into the circulatory system. So if a breast cancer patient is diagnosed with early stage of breast cancer, the 5-year survival rate is above 90%. However, if the patient is diagnosed with metastasis, the survival rate drops from 90 to 20%. And therefore, looking for the key factors or the pathway that driving metastasis is in great interest in this field. So from many talks in this meeting, that we know that the cell that grow in the primary site and enter the circulatory system, be able to survive and colonize and propagate in the distant organ requires a lot of work. And one of the important factors is the dynamic interaction between the cancer cells as well as with the microenvironment. So with that in mind, we performed gene set enrichment analysis, trying to identify the genes that associate with metastasis and the cell-cell contact, which are highly enriched in the metastatic breast cancer patient sample. And merge of the two gene sets identified 3 genes, DSG2, CLIT4, and PIM22. And DSG2 is on top of the list in both gene sets. So what is Desmoglein2? It is a component of desmosome, which is important for cell-cell adhesion. It's also important to maintain the cell integrity by interacting with the intermediate filament through interacting with the dead cell complex, including plakoglobin. So we also collect our own cohort in Taiwan and use immunohistochemistry to group the patients into DSG2 low and DSG2 high group. And the Kaplan-Meier analysis showed that patient with high DSG2 expression has worse prognosis. Importantly, these DSG2 high patients tend to have higher chance to develop recurrence. And because our sample size is really small, so we want to investigate whether DSG2 expression is really correlated with metastasis. We analyzed an independent cohort from UCSC Zena Public Hub. And we found that with this data set, the patient with DSG2 high tumor has lower distant metastasis-free survival. So from the 3 independent cohorts, really suggesting that DSG2 is an oncogene in breast cancer and is associated with metastasis. So we went on to trying to investigate how DSG2 works as an oncogene in breast cancer, utilizing a panel of cell lines for loss of function and gain of function assay. By doing sulfur-calcone forming assay as well as orthotopic xenograft model using non-singing mice, we found that depletion of DSG2 significantly decreased the anchorage-independent growth ability of the cancer cells and inhibit tumor growth. In contrast, overexpression of DSG2 significantly enhanced the anchorage-independent growth ability of the cancer cells and promote tumor growth. And since anchorage-independent growth is a very important ability for cancer cells to colonize in a distant organ, we also use these cells to perform tail vein injection, and found that depletion of DSG2 reduced the lung colonization from 100% to 28% of the animal injected with SKBR3 cells. In contrast, overexpression of DSG2 significantly increased the lung colonization from 10% to 90% of the animals that injected with MB157 cells. So DSG2 expression is important for tumorigenesis as well as colonization in distant organ. Similarly, in contrast to its oncogenic role, DSG2 expression actually inhibits invasion and migration. It's not really surprising because DSG2 is a cell adhesion molecule. So depletion of DSG2 actually increased the migration and invasion ability, and overexpression of DSG2 suppressed this kind of ability in vitro. So to test whether this is also true in vivo, we utilize MB231 cells that express EGFP with or without DSG2 depletion for autotopic model. And 5 weeks or 8 weeks after injection, we sacrifice the animal, harvest the blood. And because the cancer cell that we injected express GFP, we can just use flow cytometry to detect CTCs in the blood. So 5 weeks after injection, what we found is that consistent with the in vitro finding, DSG2 depletion really has more CTCs in these animals. But intriguingly, when we look at the blood that collected 8 weeks after injection, there's no differences between these two groups, suggests that something happened in the primary tumor that may suppress DSG2 expression. We therefore collect the tumor from this animal and stain for DSG2 and found that DSG2 expression is not homogeneous. Closer to the tumor boundary, DSG2 expression is high, but when you move closer to the center, the DSG2 protein are lower. So this is basically an inverse pattern of hypoxia in solid tumor. Whereas in the tumor boundary, there's no hypoxia, but when you move closer to the center, the hypoxic stress increased. We therefore use the serosection of the tumor, stand for CA9, which is a HIF-1 alpha target as a hypoxic marker. As you can see that in the area there's no hypoxia, DSG2 expression is high, whereas there's hypoxic stress, DSG2 protein is low. And this inverse correlation between hypoxia and DSG2 can also be observed in patient sample. This just show you 2 examples from the patient. So how does hypoxia downregulate DSG2? Well, when cells are under hypoxic stress, indicated by the stabilizing HIF-1 alpha, DSG2 gene expression was downregulated, and the proteins also decreased. And since HIF-1 alpha is a dominant transcription factor that carry out a lot of oncogenic function under hypoxia, we overexpress a non-degradable form of HIF-1 alpha and found that just with HIF-1 alpha expression, DSG2 gene expression can be suppressed. Importantly, when the cells are under hypoxic stress, depletion of HIF-1 alpha can abolish the hypoxia-mediated DSG2 downregulation, suggesting that HIF-1 alpha is important for this suppression. So further promoter analysis of the DSG2 proximal promoter identified 3 putative HIF-1 alpha binding sites. And individual mutation of this site, followed by luciferase promoter activity assay, identified that the second site, close to the minus 900 base pair upstream neurotranscription start site, could be important for HIF-1 alpha regulation. And chromatin IP further confirmed that upon hypoxia, HIF-1 alpha can bind to this site and recruit polycomb repressive complex 2 components to increase H3 ketone 7 trimethylation and therefore shut down the DSG2 expression. So to piece all the information together, we utilize 231 cells again that express EGFP with or without DSG2 depletion. And now we collect the samples 9 weeks after injection. And we also collect the blood, primary tumor, as well as the lung tissue. Consistent with our previous xenograft model, that the mice that bearing the DSG2 expressing cancer can form bigger tumor. Also consistent with the tail vein injection model, that the DSG2 expressing cancer group can have more lung metastasis. And if you look at the lung nodule, they all express DSG2. But something doesn't add up here because we know that in order for the cancer cells to invade into the blood, DSG2 has to be suppressed by hypoxia. So if in the blood, CTCs do not express DSG2 anymore, how can they colonize in the lung? So then we went on to take a look at the CTCs from the control group and see their DSG2 expression, and found that compared to the pre-injected cells, which have 98% of the cells that express DSG2, the CTC from the control animal has a broad range of DSG2 expression, about 9% without DSG2, and about 90% had low to high expression of DSG2, suggesting that either the cell somehow can re-express DSG2 again in the blood, or maybe those DSG2 expressing tumor cells on the tumor boundary can actually collectively move to the blood. We therefore use a chip system to capture CTCs, and indeed found that, well, the single CTC doesn't have any difference between these 2 groups 9 weeks after injection. However, if you look at the CTC cluster, the control group, meaning that the animal that bearing the DSG2 expressing tumor has more clusters. So whether DSG2 can be re-expressed in the blood. Just to prove of concept, we put the cells into the hypoxia chamber and move it back to normoxia, and indeed found a de-repression of DSG2 expression. So combined our clinical data, animal model, as well as cell-based assay, we're able to propose this model that in breast cancer tumorigenesis, in the primary site, DSG2 expression is important to facilitate anchorage-independent growth ability of the cells and therefore promote tumor growth. And when tumor is big enough to generate hypoxic stress, hypoxia will induce HIF-1 alpha to recruit polyclonal repressive complex 2 to suppress DSG2 expression and allow cells to go into the bloodstream. And once in the blood, the hypoxic stress decreases, and it can probably re-express DSG2. But from Daniel Haber's talk, we know that it's probably a really rare event. So there's alternative possibility is that the cells that have DSG2 suppressed by hypoxia, once they move into the bloodstream, they actually go to the lung and then colonize there. And in the lung, the hypoxic stress is not as significant as in the primary tumor, so DSG2 can express, and then they can start to propagate. And of course, there's another possibility that actually the tumor cells that close to the boundary that express DSG2 can move out of the primary site collectively and colonize in the distant organ. No matter which route it goes, our study, I think, is really showing that DSG2 is very important oncogene in breast cancer. With that, I would like to acknowledge the person who did the work since the lab started 3 years ago, talented Howard Kim Bo-Hao Zhang, a postdoc, and also the collaborator, the pathologist in National Taiwan University Hospital, as well as the funding sources from Academia Sinica and the Ministry of Science and Technology in Taiwan. With that, thanks for your attention. I would like to take any questions.
Thank you. So, junior scientists first. Celeste.
Guess I'm a senior scientist. So I might be really behind on my reading, but that's the first time I've ever seen evidence of HIF-1 alpha actually recruiting negatively acting cofactors. And I'm just curious if you've been able to re-chip at that site, or is there a possibility this is all acting through HIF-1 targets, which are things like the KDMs as a good example. So are you guys really seeing at those HREs direct interactions by things like re-chip, or could there be sort of some extra steps in between, I guess is my question.
We are doing some Biotin probe pull down assay using the wild type sequence versus the mutated sequence of that promoter region. And we can really see that HIF-1 alpha cannot bind to the mutated site. We're working on the re-chip as well, and there's also another possibility that those polyclonal repressive complex components are actually regulated by HIF-1 as well. So is that also possible that is a general hypoxic effect? We're still trying to figure it out, but definitely re-chip is a very good way to deal with that. Thank you.
Yeah, very nice. So
Thank you.
Two quick questions. One is, this clustering phenotype or this pro-metastatic phenotype, do you think it's mediated via the desmosome, or is this a unique function of DSG2?
There's a paper published saying that the plecoglobin, which is one part of the desmosome component, is also involving the collective migration of the CTC clusters. So it could be through the desmosome function. What's more interesting to us is that how DSG2 acts as an oncogene in those primary sites that only depending on the desmosome cell adhesion function or something else. And it's under intensive investigation in the lab now.
And you showed that in the circulation you get, I guess, a re-expression of DSG. So a lot of times when you matrix detach cells, they tend to increase a variety of cell-cell adhesion pathways. And I'm wondering if you've seen any direct direct relationship between loss of matrix attachment and increase in DSG.
We haven't looked into that, but definitely some aspect to look into. Thank you for the suggestion.
So is it just about tumor pressure coming out into the circulation, or is there more to it, like better survival in the lungs, or
we think
better
it's
growth?
probably pro-survival as well. But how exactly that works, we don't know. We're looking into our mass spec data, trying to see whether there's other pro-survival transmembrane protein that can interact with DSG2 or any protein molecules that in the cytoplasm area that interact with it, but still working on it.
Okay, thank you.
Thank you very much.